So in this lecture, we're going to be talking about how proteins and other macromolecules move around eukaryotic cells. So how do they move around in the cytosol and how do they move to different organelles? And in this first lecture, we're going to be focusing on nuclear localization signals and how proteins move in and out of the nucleus. So to start with a big picture, the goal of trafficking is to put things where they belong in the cell. And so to do this, every protein or macromolecule may need a zip code to direct it to a particular location. In today's class, we're going to be talking about proteins that get to the nucleus or come out of the nucleus. Another analogy is that each one of these processes needs a mail carrier to be able to read that zip code. And in some instances, we're going to need a mail slot. And this is a protein or a complex of proteins that's going to allow us to cross over membranes. And this is going to take energy. And so we can think of this as a delivery vehicle that is bringing the carrier from place to place, and that delivery vehicle needs gas. So these are just some analogies to keep in your mind. And in each example, when we talk about proteins being trafficked, I'm going to point out the zip code, the mail carrier, the mail slot, and the energy investment. So try to keep track of those as we move through, right? In this lecture, we're going to be talking about the nucleus. And then in the next lecture, we'll talk about the endomembrane system. So let's consider what needs to move in and out of the nucleus. So we've briefly talked about the central dogma. And the first step of the central dogma is when DNA, which is a double helix shown down here, gets converted into mRNA. And this process is called transcription, and it happens in the nucleus. The next step is that mRNA is converted into protein. And this can happen in the cytosol, but also in the cytoplasm. So it can happen in the ER. And this step is called translation. And we talked about how the process of translation, converting mRNA into protein, requires ribosomes. But ribosomes are made in the nucleolus, which is in the nucleus. So that means that assembled ribosomes and mRNA, right, which were all generated in the nucleus, actually have to move out of the nucleus to be able to help with translation. So those are things that need to move, just some of the things that need to move out of the nucleus. Let's consider what needs to move into the nucleus. So recall that ribosomes are made up of, that should say, rRNA and protein. And so that protein is actually made in the cytosol. And so that protein actually needs to make it back into the nucleus once it's made. For this process in transcription, where we convert DNA into mRNA, this requires several enzymes that we call transcription factors that help us read and transcribe what genes need to be made in the cell. And so these can be made in the cytosol and then need to be brought into the nucleus. During cell division, DNA will need to be copied. So we'll need 2 copies of DNA to pass along to daughter cells. And this process requires DNA replication enzymes. And so that's another example of something that needs to be brought into the nucleus. And so thinking about what proteins, just a general description of what proteins might do, would give you a clue about whether or not they need to be brought into the nucleus or come out of the nucleus. So just to review, things that might go in include transcription factors, ribosomal proteins, DNA replication enzymes, but also building blocks like nucleotides to make DNA. And there's a lot more that I'm not covering, but things that need to move out of the nucleus, so these are things that are made in the nucleus, include assembled ribosomes, as well as mRNA and other products. So how is this going to happen? So let's walk through the different steps, keeping in mind, right, we need a zip code and a mail carrier and some sort of energy, as well as a mail slot. So I want to remind you about some of the features of the nucleus and its mail slot. So the nucleus is large and it's highly organized, and I'm going to point out some key regions. So we have the nucleolus. This is the site of ribosome assembly. We also have underneath the nuclear envelope is the nuclear lamin. This is made up of intermediate filaments, and chromosomes are anchored to the nuclear lamin, and it helps provide structure to the nucleus. And then on the outside is a double membrane we call the nuclear envelope. And you can see in this cartoon here that this is a double membrane, and the nuclear envelope is also studded with these green circles here. These are pore-like openings, okay? And we're going to zoom in on what those pore-like openings are. So the nuclear envelope is perforated with openings called nuclear pore complexes, and that's shown here in this transmission electron micrograph. This is the double membrane of the nucleus, the nuclear envelope, and you can see there's this contact point here. And if we were to zoom in on this, we would see this very large protein complex made up of approximately 30 different proteins, and this is a cartoon of what it might look like. And so it crosses both the outer membrane and the inner membrane of the nuclear envelope. And it creates this pore-like structure where we have, over here is a cytosol, and we have some proteins that can interact with proteins in the cytosol. We have structural proteins that form the pore and this basket. And so this creates a path for proteins and building blocks of DNA and RNA to come in, and ribosomes and mRNA to come out. So that's our mail slot. So how do proteins get into this mail slot? What's the zip code for the nucleus? The zip code for the nucleus is a 17 amino acid long nuclear localization signal, or NLS. So this is how you would address something or a protein to the nucleus. And this serves as a zip code to pass through that nuclear pore complex and allows proteins with an NLS to enter the nucleus. And the NLS was discovered in this experiment here with this protein called nucleoplasmin. And this was a nuclear protein, and it had two parts. This protein was a complex, but it had a core, which are represented by these balls, and tails. And these are proteins that have an N terminus and a C terminus. And what they did in this experiment was that they used a protease, that's an enzyme that clips apart proteins, to separate the tails from the core. And so they had separate parts of this protein, and they labeled both parts with radioactivity. So that's shown here in these yellow stars. So they labeled them, and then they injected those proteins into the cytosol of different cells, okay? And what they were looking at is what part of this protein contains the zip code? What part of this nucleoplasmin has the nuclear localization signal? Is it an amino acid sequence within the tails, or is it an amino acid sequence in the heads? So if they predicted that it was in the tails, then we would expect only the tails to appear in the nucleus rather than the whole protein. But if the nuclear localization signal was in the head, then we would expect these labeled proteins to end up in the nucleus. And so their results showed them, or supported the hypothesis, that the nuclear localization signal was in the tails, because all the labeled tail fragments located to the nucleus, but all of the core regions actually stayed in the cytoplasm. Even though this was a nuclear protein, those no longer had the nuclear localization signal, and so they couldn't enter the nucleus. So these were the first experiments that demonstrated the presence of a nuclear localization signal. And let's talk now about how exactly this nuclear localization signal helps proteins get into the nucleus. So let's talk a little bit about nuclear transport. So here is a cartoon diagram of our nuclear pore complex. And so you can imagine this is actually inserted into the nuclear envelope, and it passes through both the outer membrane and the inner membrane. Now, small proteins and small metabolites can cross through these nuclear pores without any zip code or input of energy. And so there's an example of a small protein crossing over this nuclear pore complex. And this process doesn't require energy and it can happen spontaneously. But large proteins can't easily fit through this complex. There's a mesh-like network in here that basically prevents this large protein from entering. But if this is a large protein, let's say a ribosomal protein, it actually needs to get into the nucleus so that ribosomes can be assembled. And so that's where the nuclear localization signal comes in. And so I'm showing the nuclear localization signal as this blue bar here. And so you can imagine that this is a protein, right? And this is a part of the protein that sticks out. And it's an amino acid sequence, a 17 amino acid sequence. That's the nuclear localization signal, the zip code. And so this is our zip code. Here's our mail slot, the nuclear pore. And we're going to have the input of energy, and we're going to come to this in a little bit, in the form of GTP. And I talked about GTP very briefly when we talked about tubulin. But I just want to show you that GTP is pretty close to ATP. So ATP stands for adenosine triphosphate. And so here's the adenosine, the sugar, and 3 phosphates. GTP is guanine triphosphate. So here we're switching out our base, but we still have our high energy phosphate bonds. So GTP can work like ATP in terms of energetic coupling. So I'm going to introduce you to this protein called importin. And importin is a protein that has affinity for nuclear localization signals. That means it's going to bind to other proteins with that sequence, with that 17 amino acid sequence. And importin is going to act like the mail carrier in this example. So it's going to bring the mail, our nuclear protein, and it's going to pass it through the mail slot. So that's what that would look like. Okay, so now we've brought our large protein with the NLS into the nucleus, but it's still bound to importin. So it can't do what it needs to do. It's stuck on another protein, and this importin needs to be released and recycled back out of the nucleus. So this is where RAN-GTP comes along. And RAN-GTP is this protein that's called a G protein. And there's a lot of different G proteins in the cell. And what's unique about a G protein is that it is on or active when it's bound to GTP, and it's off or inactive when bound to GDP. So when RAN-GTP is in the nucleus, it's on, and when it encounters importin, it has high affinity for importin, and it outcompetes this nuclear protein with the nuclear localization signal. So it changes the conformation of importin and releases this nuclear protein. It also lets importin move out of the nucleus. And once out of the nucleus, RAN-GTP hydrolyzes its GTP to become RAN-GDP. And so it's no longer active, and it dissociates with importin. And now importin can pick up cargo and repeat this process all over again. Nuclear export basically is the same pathway, but happens in reverse. And I'm not going to go over the steps of this pathway, except to point out that large proteins can have a nuclear exit signal or nuclear export signal. And these proteins in this nuclear export signal interact with a protein called exportin, and this has affinity for the NES. And so these bind, and then exportin can take this protein out of the nucleus, and RAN-GTP can help release exportin from these proteins with an NES. Okay. So to summarize, nuclear localization signals act as zip codes to bring large molecules into the nucleus. These are stretches of 17 amino acids located within proteins. And proteins that need an NLS include transcription factors and enzymes that function in DNA synthesis and replication. We also need to bring in nucleotides, which are small molecules. Nuclear export brings molecules out of the nucleus, including ribosomes and things like mRNA. And this process requires, for the large proteins and molecules, requires energy in the form of GTP. So that's all I have to say for this video, and then we'll talk about proteins moving around in the rest of the cell in the next video.